I'm a neuropathologist by profession, and as a neuropathologist we are interested to understand how the immune system in the brain fights diseases. And parts of the immune system are tissue macrophages. You find tissue macrophages in all tissues of the body, and inside the brain these are microglia cells. And we now said these are specific macrophages that live for a very long time, for years, and we would like to understand how these tissue macrophages fight with disease and what's the influence basically of the host microbiome on these microglia cell function is. As I told you, microglia cells are tissue macrophages, and these are not only big eaters of any debris, but they're also disease modulators. And what we know from different studies from different labs, that microglia cells are participating in any pathology in any disease. If you have Alzheimer's disease, if you have multiple sclerosis, brain tumor, microglia cells are active in any kind of pathology. And researchers, including ourselves, we want to understand how we can instruct microglia cells to fight the disease. And host microbiota, as we and others have shown, are important mediator of microglia function, fighting the disease. What we have found a couple of years ago, it was already 2015, was quite surprising, because the whole brain is protected by the blood-brain barrier, and we consider the brain as an immune privileged organ. So it means that bacteria, for example, have no easy access to the brain. Otherwise, you would have a brain inflammation, abscess. So that's why we were quite surprised when we saw that in the absence of host microbiota, and we used for this investigation so-called germ-free mice that have never seen, were never exposed to host microbiota, then these germ-free mice, the immune compartment of the brain, that means the microglia cells, are strongly impaired. This was totally surprising, unexpected, and started a couple of experiments in the subsequent years to come. To understand how the microbiome contributes to microglia function and modulating of brain disease, we used either normal SPF mice that have a normal repertoire of host microbiota, germ-free mice that have no bacteria at all, no host microbiota, and antibiotic-treated animals. And as I told you, in the antibiotic-treated animals and the germ-free mice, microglia cells are quite immature and cannot function normally again. And if you ask what are the difference between antibiotic-treated animals and germ-free animals, we found that microglia function is changed in a different way. Yeah? But under both circumstances, microglia cells are still immature and dysfunctional, fighting in a different way and a less proper way to CNS diseases. And we are currently trying to understand what is the difference if we deplete microbiota by antibiotics just for 4 weeks, or if these mice have never seen any antibiotics like in the germ-free mice. One of the major message of our study was that we were able to restore the immature phenotype in microglia cells seen in germ-free mice, because we repopulated germ-free animals. That means we gave them back the normal host microbiome. This took about 4 weeks. And we were quite surprised and impressed that this transfer of the normal host microbiome was able to restore, at least in these animals, in these mice work, fully functional microglia cells, indicating there's a highly interactive and plastic process between the host microbiome and the CNS immune system. I think this is certainly the big hope for the future, to understand how we can precisely modulate CNS disorders, not only in mice, but also in humans. And I think there's much work to be done, certainly, because we are just starting to understand there's a vital interaction of both compartments. And what you have to keep in mind if you, for example, give short-chain fatty acids or specific bacteria, this reacts not only on the brain, not only on microglia cells. The whole body is responding. Many cells are responding, and even in the brain, not just the microglia cells and other cells as well. That means it's quite complicated scenario, but certainly I think the wish for the future to fight diseases with specific microbiota. Our results were very surprising, because people thought before that the brain is totally protected. It's really totally immune privileged. That means there are no signals coming, no bacterial signals coming during homeostasis for the normal brain from outside the brain. And That when we saw that in germ-free animals, microglia, macrophages are totally immature, this was not only surprising for us, but also for the community. And that we could further show that it's a highly interactive and plastic process, modulating macrophage function in the brain and thereby modulating disease. This was a groundbreaking discovery of our research. As I told you, microglia cells are involved in virtually all CNS disorders, ranging from neurodegeneration like Parkinson, Alzheimer's, but also CNS tumors, but also inflammatory disorders like multiple sclerosis. And as a matter of fact, we are also working together with clinicians, neurologists, who are treating patients with specific short-chain fatty acid receptors to modulate the cause of multiple sclerosis, and to modulate by giving short-chain fatty acids the immune system peripherally, as well as in the CNS, like microglia cells. I think for the future, for the perspective, many studies will be undertaken to understand how host microbiota, how short-chain fatty acids modulate the immune system inside and outside the CNS.
Yeah, so my presentation was on social brain, how the microbiota influences social behavior. And while a lot of our current understanding about it has come from germ-free mice, which have been an incredible tool for the field, they do come with their own limitations. Germ-free mice have profound alterations to their physiology, both in the rest of their body and indeed in their brain. And this kind of results in profound behavioral changes, not just in social behavior. They have alterations in anxiety behaviors and their response to stress. They're perhaps not as translationally relevant, or they can only answer the question up to a certain point. So then, there has been some work by our lab and others in kind of identifying the mechanisms in a normal mouse, how gut bacteria communicate with the brain. So Rich Ellis yesterday gave a talk on bacterial peptidoglycan, which is a component on the bacteria, and that can signal to the brain. And there's been work done by others to kind of see, with Lactobacillus reuteri, so this is a very kind of popular probiotic in the field, and it's used quite routinely with preclinical studies. And this has been shown to increase oxytocin in the brain. And oxytocin is a pro-social hormone. It's very much linked to autism. There's alterations in this hormone in autism spectrum disorders. So this strain has been shown to increase Lactobacillus reuteri via the vagus nerve. So the vagus nerve goes from our brainstem all the way down to our gastrointestinal system. And what happens is, if you sever this nerve, then the bacteria in the gut actually can't communicate with the brain, and you don't get this increase in oxytocin, and subsequently you don't get this change in social behaviors. There's certainly a lot more that we need to know, because there's more than one or two strains in the gut microbiota. There's millions. So the relationship between how gut bacteria communicates with the brain in terms of social behavior, there's certainly a lot more that we need to know that we don't presently know. The field itself, in the context of neuroscience, largely focuses on mice and rats to model what we see in humans, and certainly they're an invaluable experimental tool. But, and I suppose our research to date has been focusing on, autism spectrum disorders and modeling autism spectrum disorders in mice. And, it might be easy to forget that this relationship between gut bacteria and social behavior is not limited to just mice and the translational relevance of what we see in humans. It's actually observable across the animal kingdom. So in my talk, I said we can see it in invertebrate species, very simple invertebrate species like ants and termites, all the way up. So you can see it in birds, you can see it in species like hyenas, koalas, and even in non-human primates. So the example that I was talking about was locusts, for instance. They produce a chemical from their equivalent of the gastrointestinal system, and what that does is this chemical mediates attraction. So it acts as a pheromone. It attracts all locusts in the surrounding environment to come together and mediate social cohesion. And so this is one example of how the gut microbiota can do this. And then hyenas, there was a very interesting study a few years ago with striped and spotted hyenas, so two different species. And what they noticed was that the paste that is produced from the scent gland of hyenas differs between social and non-social hyena species. So the social hyena species produces a paste that is very diverse in gut bacteria and kind of volatile acids that give off a lot of odors, whereas the non-social hyena species produces a paste that doesn't really contain a lot of bacteria, or less than in the social. And what these people theorize is that the social hyena species actually uses this complex paste as a means to communicate with other conspecifics. Yeah, so my plans next, I've been working with a kind of live biotherapeutics or a strain, to improve autistic-related behaviors in mice. And we've seen some very interesting behavioral changes. And I think what's quite unique about this particular strain is that it's capable of improving all kind of aspects of autistic-related behavior in mice. So as I was saying in my talk, we kind of broadly refer to autistic-related behavior in mice as being a deficit in social behavior, increased repetitive or stereotype behaviors, and heightened anxiety. When we see the behavioral deficits in humans who have autism, it's a lot more complicated and diverse. But for the sake of our preclinical studies, we kind of just focus on these 3 core behavioral deficits. And we've seen this with this particular strain that's capable of improving all of these 3 core behavioral deficits, but really we don't know how it's doing this. And I think it'd be very important to delineate its mechanism of action, which is easier said than done because it's very much like a needle in a haystack. We don't know how it's doing this, whether it's altering the microbiota, whether it's releasing something. That's something that we're going to have to investigate in the next few months.
What do you think about the conference?
I certainly think there is an added value. The conferences that I've been at so far where I've presented microbiota data have been largely neuroscience conferences. There was always an interest in the data that we were presenting, but it was quite general meetings, whereas this has been fascinating because while the microbiota field is expanding, I think this microbiota gut-brain axis, it's kind of one aspect of this field. And this meeting has been brilliant, bringing all these people together, people who I've read their papers countless times and I kind of hold them in high regard, and it was a little bit starstruck meeting them nearly, but they're lovely people, and I think it's a great opportunity for all of us to kind of communicate our ideas and work together, because I think, in the future, the field of the gut-brain axis is really going to have to focus on elucidating mechanisms of action. So bringing us all together like this with mind, mood, and microbes is a great opportunity to give us the opportunity to discuss how we're going to approach this. Does the microbiota determine how social we are? I don't think so. I don't think there's enough evidence in humans to really suggest to say yes or no. There's a lot of other factors that determine how social that we are beyond our microbiota. So, our genetics plays a very crucial role in that. And I think it's too premature to say yes or no. And we can certainly see it in animal models and across the animal kingdom because they're a lot easier, I suppose, to investigate, and their behaviors are not as complex as humans are. So in order to infer that in humans, which, we're quite evolutionary complex species, I think it's going to take a lot more work. But I'm just happy now to open up the dialogue about this concept and see what the field can do with it down the line.
All right, so if I would explain it to someone like my mother or my brother, I would say that the talk was about how the gut bacteria, the microbiota gut-brain axis, or how gut bacteria affect the brain and human behavior. And I'm specifically studying how that affects human behavior. So how we can measure that, how, for example, do people recognize other people's emotions, or how do they make decisions? And in order to investigate that, I'm specifically looking at the direct physical link between the gut and the brain, which is called the vagus nerve. So it's a direct physical link, a nerve that connects the gut to the brain. And we can either measure its activity or activate it by applying electrical stimulation and see how that changes your behavior, for example. So I think that all of us scientists that are here at this conference, we all ultimately want to know how this gut bacteria, the microbiota, how they affect the brain and behavior. And we do that because we all have this sort of aim of helping people in preventing disorders or treating psychiatric illnesses or things like that. And of course, it's very, very important to do the animal studies, but ultimately we have to start looking at humans also. And I look at healthy humans because we actually know that there are some things that healthy humans show that can make them be more at risk, for example, for developing something like a depression. So if you are, for example, at this moment you have no complaints or symptoms that look something like a depression, then still it may be that you ruminate a lot, you overthink your life, and that is actually a risk factor. So if I can see how the brain, how the microbiota affect this rumination, this overthinking, then perhaps we can already start using that to, treat or prevent things like depression. Yeah, so the vagus nerve is the direct physical link between the gut and the brain. So basically the vagus nerve is the nerve that transports the information from the gut to the brain. It's one pathway, there are many more pathways, but it's the most, let's say, direct pathway because it's what we call a neural pathway, basically an electrical pathway. The signals go very, very fast. And what we see is that, for example, diversity in the gut bacteria can be related to activity of that vagal nerve. Or some probiotics, so adding some bacteria to your gut can change activity of the vagal nerve. And we've seen that in animals. We've also seen, for example, in animals like mice, we can cut the vagal nerve, so we can cut the pathway and then the microbiome or probiotics show no effects on behavior anymore. But we don't know yet how that is related to humans and especially healthy humans. So that's why I'm looking at the vagal nerve. We know that it's one of the key pathways in bringing the information about the gut and from the gut to the brain, which affects your behavior and your feelings, but we don't know how yet. Yeah, so let's say the most of the evidence we have is very indirect at this moment. So we know that, for example, activating the vagal nerve can help in treating depression. We also know that lower what we call heart rate variability, so that's activity of your vagal nerve, is actually related to things like overthinking, rumination, and higher heart rate variability, which is again a measure of this activity of the vagal nerve. So higher activity of that is related to more mental flexibility as well, so less overthinking. So still, we don't know exactly how, but what we see at least is when we activate the vagal nerve, people become better at recognizing emotions, for example. And we know also that people who suffer from depression are worse, especially at recognizing positive emotions. So it seems to do something, this vagal nerve and activity of it seems to do something with emotions, with overthinking, with basically abilities and functions like that. But again, we don't know how yet. So by a psychobiotic, we basically mean a probiotic or a prebiotic, a bacterial, either a bacteria or like food for a bacterium that affects your mental functions and well-being, for example. So when we're talking about probiotics, like in my studies or in my field, when we're talking about probiotics, we're talking about bacteria and probiotics in relation to mental functions, which automatically makes it a psychobiotic. I think at this point also, my aim basically is to help people in reaching their optimum in healthy functioning. So help people support their healthy functioning, and that can be used maybe to prevent disorders or to enhance their abilities, things like that. But you could question, for example, whether an antibiotic might also be a psychobiotic. So if we define a psychobiotic as something that has a positive influence, we now see that sometimes antibiotics can actually be used to decrease depression symptoms. So I would not necessarily say we should all start using antibiotics. On the contrary, but an antibiotic could also be a psychobiotic. The most difficult thing about these probiotics in relation to mental health or mental functioning is that there's not one, probiotics is not one thing. We have many different mixtures and bacteria, so which one should you use? I think the practitioners are ready to start trying it by means of, let's say, a pilot, because there is evidence showing, I think there's now enough evidence showing that there might be potential, there might be beneficial effects. So from my perspective, I think they should be ready, but from their perspective, well, they might not necessarily agree. I think, though, that the patients or the people at home are ready, but they need to be guided in trying this. So if you're a depressive patient, for example, maybe there are actually some studies now showing that adding probiotics to your antidepressive treatment might actually speed up the process of recovery. But of course, that doesn't mean that you should stop taking your medication at all. And definitely your psychiatrist or your clinician should help you guide basically in doing that. But that does mean, of course, that not only you as a patient need to be ready and willing, but your clinician or your psychiatrist needs to be as well. So I hope they will join the crowd soon. What I like about this conference most is that it really, really brings together the pioneers of the field, but also the clinicians, the companies. So we are really able here in Amsterdam to basically put our heads together and to start thinking about the next steps. So not only is everyone presenting what they've done in the past and what we see, what kind of developments we've made in the past years, but also we put forward the next questions. And the nice thing is that immediately then after that, over coffee, you can actually start discussing how we can start answering those questions. Because of course, a field like this in the microbiota gut-brain axis field, we need people from microbiology, from gastroenterology, from neuroscientists. We need psychologists like me, I hope. And we need to bring them together to answer this question of how the gut is affecting the brain. So that's what I think the value of this conference, and especially because they're all here.
Well, basically, I do a lot of research in the obesity field, especially in kids and what's happening in their at the physiological level during weight loss. And I have this other field where I also work with probiotics. And but the field of gastroenterology and stress physiology is especially one of the topics of my boss Paul Enck, which is not here unfortunately today. So I presented today also quite a lot of studies from him, conducted from him and students. Yes. Well, what I like to do is let's have a look at IBS patients first. We had conducted a systematic review in 2015, and there were 56 studies included with probiotics, RCTs and probiotic studies conducted in IBS patients. And what you could see is that the multi-strain probiotics weren't really useful. And at the single strain level, you really had to be specific. So
why were the multi-strain not useful? Just because of the setup of the trial?
Or well, I mean, of course, the trials are very heterogeneous, and also you have different subtypes of these IBS patients. Often it's mixed studies, right? But with the multi-strains, you have to look what you're doing there, because if you have a single strain, then you know this single strain will exert these or these effects. If you do combinations, you don't know whether they inhibit each other or whether they have promoting effects together. So you have to be specific, and this is also where I think it's important for the future to really look what are the single strains doing, and can you use them in combination? Yeah. Right. And so I think in the past there was it were commercial interests, or you looked like which kind of probiotics could you easily culture. Many are from just from the yogurt industry, where you have Lactobacillus lactis or something, and so it's a history of these multi-strains.
It's not from Yakult.
It's this history, right? And I think the future is more to be really specific, to look at the and also go down to the strain level to be sure which strain is doing what. Because like you and me, we're different, right? And that's basically what the strains are when you look at the probiotics. It's the you and me.
So what was the efficacy considering IBS coming out from your
Oh well, I don't want to go into the specificity, because it's not. I forget all the strains which worked or which did not work. Generic, but there's some where you have more negative results, and some there where you have positive results at the single strain level. So you have to be specific and look.
Did you prepare a force plot, for example, that you can at least see an overall effect? Or?
In this. I haven't done this systematic review myself, but it was in this case not useful, but another study which came out later, they did it then.
Okay.
Right. Okay. And now, of course, we moved forward. We are looking at probiotics and CNS function. There is quite a lot of studies in mice using different stress paradigms, starting from early life stress to chronic unpredictable stress to acute stressors. And most of them use lactobacilli or bifidobacteria strains. And overall, you find that anxiety, depression improves, or memory function as well. There is 4 studies published at the moment which really look at CNS function in humans using different probiotics, and they use these facial, emotional facial expression tests.
Mm-hmm.
Most of them show positive results. There's one which didn't show anything. And we have conducted an fMRI study now using Bifidobacterium longum 1714, using a stress paradigm, the cyberball game. And we could show that it has stress reducing effects, and the results were so convincing that it's now the first patent pending probiotic, a precision biotic, which we're going to use against stress. And there is now also some studies going on as well with this, some further studies where we're using the Bifidobacterium 1714. So let's see what is coming out.
Yeah. And what are possible pitfalls in research considering this field, and of course also promising directions?
Well, I think first it's that we always have to think about the study designs. They have to be appropriate. We must look that if we go into patients that they're not too heterogeneous, we should stay specific. And this is also what I think what we have to consider using probiotics working on which act on central nervous system function to be specific, and not to do all the mistakes which we have done over the last decades in IBS research or in other gastrointestinal disease, to be really specific to know what is going on.
Oh.
I think it's a very nice venue here, and it's extremely good, it's extremely well organized. And I just can say thank you. It's people here from all kind of disciplines. And I like that, and everyone is very open, and this is nice. So I would like to attend the conference again. So I would be happy if the society is keeping going.